Hi guys, welcome back. Now, as time is catching up with us, I'm going to try and go as quickly as possible. Now, one thing I've missed out in my series of videos so far is the transmission of diseases. Okay, so in your revision, the very basic idea is that pathogens are organisms that could enter another organism and cause harm to that organism. And in that context, we are looking at three infectious diseases, okay? Now, what that means is they can travel from one person to another, causing harm in the infected individuals. Now, the three infectious diseases that you need to know about are tuberculosis, HIV, AIDS, and malaria. Okay? Now, it might help you to understand or make a link with what type of organisms there are. Remember, we're always trying to make links with other parts of the course. This is a bacteria. This is a virus. This is a protoctist. Okay? Now, what I want to look at is essentially, there's 4 points here, but essentially there's only two things I really want to discuss. One, well, let's just look over the four things first of all. Each of these organisms has to enter the host. They have to reproduce. They have to cause damage to that host. And finally, they've got to travel from one host to another. No point killing your host before they have a chance to pass on the disease from the pathogen's point of view. Okay, now, if you can go through that list and you can, quickly review for each of these pathogens, these points, if you can explain each of these points for each of these organisms, you're okay. But you should be asking yourself questions, or you should be thinking about doing more work on this in this area if you can't explain how each of these organisms does each of these things. All right? Now, for me, I would break this down to two things. Okay? I would break it down to two things. Shall we call it three? No, let's call it two. Okay, two things. Let's try and make it simpler. So, one, how does the organism cause damage once it's in the host? That's one thing you really need to have clear in your mind. And secondly, how does it get from one host to another? Okay, so that is transmission. So let's call this pathology, the study of how this microorganism is actually going to cause damage to the host. And the second part is transmission. All right, now I'm not going to focus too much on transmission because that's simply just bullet points from the textbook. You just need to go through them and make sure that you know them, not much more than that. I would also suggest that you look into the kind of socio-economic factors that can affect the transmission of these diseases, as well as things like climate. Yeah, so these are all kind of the things that factor into the spread of the disease. All right, so today I'm just going to focus on the pathology. The transmission follows on from that, but that I think just comes down to revision. It's the pathology that I think might need a bit of trying to get your head around, but that's why I'm here. But once we've done that, hopefully then things should be more straightforward. All right, so let's begin. Hi guys. I know I said I wouldn't talk about transmission too much because it was essentially just going through the textbook and noting down the appropriate bullet points. But something you might find helpful, I don't know, is this, that there are three main kind of ways that transmission can occur, and they are listed in the textbook. but I thought it might be kind of interesting to just make a note of those three ways. And the three examples of infectious diseases that you need to know about all are transmitted by three different ways, which are referred to in the book. So the bacteria is transmitted via droplet infection. Okay, so coughing or sneezing, that produces droplets of mucus from an infected person, contains the pathogen, which then can spread from one person to another if someone sneezes it out or coughs it out, and then those microscopic particles get then inhaled by someone else. Virus. So in this case, the HIV can be transmitted by actual physical contact. Now remember, this is not all viruses, it's this particular virus. Cold and flu, droplet infection. Okay, HIV, physical contact, in this case sexual intercourse, and so on. Okay, so that's physical contact, that's the second means of transmission. And finally, malaria relies on a vector. So this is another organism which will be transmitting it from one human to another human, not by sneezing or not by touching or sexual intercourse, but by the means of the other organism, in this case being the mosquito. All right, so vector, relying on a vector organism. Okay, so I found that quite interesting. Hopefully you might find it helpful to kind of, if you want to differentiate the three different ways, the three different diseases, there are these things as clues. Okay, right, now let's get to the real stuff, the pathology, next. Okay, so let's look first at tuberculosis. The first one. Alright, now remember, tuberculosis is simply a bacteria. Okay, kind of has a shape like that. Alright, now remember, bacterial cells are very much smaller than animal cells, okay, or eukaryotic cells. So just bear in mind this size difference, okay. Now, the textbook is a little bit vague on tuberculosis and how it's working, but understandably so, because there's not really too much to this. So you have your lungs, right? And we've spoken about the lungs before. All right. Now let's say there, right? It's alveoli, bronchiole, bronchus, trachea, etc. Okay. And through a droplet, someone could inhale the, a very small droplet of mucus which contains, lots of tuberculosis bacteria. Right, now that would go into the lungs and make its way in between the cells of, that make up the walls and the tissues of the lungs. All right, so we're not talking about going into cells here. All right, so we're looking at the kind of cells that make up the walls of the bronchioles, maybe the endothelial cells of the epithelial cells of the alveolus, etc. Okay, so we're just looking at those cells, and we're kind of considering that maybe in between those cells we get a bacterial cell of tuberculosis, M tuberculosis, for example, Mycobacterium tuberculosis. All right, so we get one cell kind of making its way in there. All right, now first thing, yes, we have the ciliated epithelium and we have the mucus, and that's all well and good, and it's doing a good job kind of removing dust particles and pathogens. However, the first issue with the tuberculosis and how it can cause infection is by possibly getting through those defenses. All right, so those defenses are there doing a job, most of the time, but it could get through. So what you have to accept that first of all is that some tuberculosis bacterial cells get through what we know, what we call our primary defenses. Okay, and this is happening, remember, in the lungs. Okay, so what happens is, it gets through here and it ends up in between the cells of our important tissues. So let's just zoom in. We have a few cells here. All right, and that's some cells of some tissues of the lungs, and in between that we've got our little bacteria, and you might not be able to see that because bacteria are very small. Anyway, so what can happen is, these bacterial cells, this is tissue fluid. Remember tissue fluid? Tissue fluid is in between these cells, and remember tissue fluid has got high in nutrients because essentially tissue fluid contains all the nutrients that all these cells need to build their own proteins, lipids, nucleic acids, etc., etc., glucose for respiration. And surprise, surprise, that's also what the bacterial cells will need for them to build their own biological molecules, and so therefore build their own cell materials before they can divide. So in the tissue fluid they can get all the stuff that they need, and therefore they can multiply. And as they multiply, they will produce chemicals, substances that attract our secondary line of defense, our neutrophils and macrophages. All right, so once in between the tissue cells, so once they get through the secondary primary defenses, they will multiply and essentially attract phagocytes. Okay, so they will multiply, they will attract phagocytes into the area. Okay, so our phagocytes, they are attracted to some of these substances that are, released by the bacterial cells. The bacterial cells are not doing this on purpose. It's just a kind of byproduct. Just like our cells can't help releasing carbon dioxide, in the same way the bacterial cells can't but release some of their products, just a natural process of their survival. But our secondary defenses recognize these things and recognize that there's likely to be an infection, and so move towards the source of those chemicals and move towards the tuberculosis bacteria. So here they do kind of neutralize to a certain extent the bacteria, and once, as we've done the immune system already, so the phagocytes act, so 3 phagocytes act as antigen presenting cells, and they activate the adaptive, the specific immune system. So phagocytes, they activate the immune system, and so our body makes a directed immune response to these cells. And what happens is that the bacteria eventually get encased in many cells of our immune system. All right? Effectively, they're sealed off from the rest of the body. Okay? So we have our, maybe I should just highlight them. All right, so we have our bacteria there, phagocytes, activate the immune system, and eventually we have them all encased in the immune cells. Right, now these structures are called tubercles. All right, so for the bacteria become encased in immune cells, okay? Lymphocytes, phagocytes, they become surrounded by them and in a structure called tubercles. Tubercles, and these are visible under the microscope, hence probably why the disease, the bacteria, was named as, tuberculosis. Okay. Okay. So they become encased in these tubercles, and actually, what can happen is that the disease at this stage can just kind of stop. All right. So these bacteria are still viable in there; they're still alive, but there's this kind of balance. So they're multiplying, but then your body's responding with an immune response, multiplying immune response, and that kind of reaches a kind of equilibrium, all right, where nothing much is going on, but there's constantly, your immune system is constantly working to kind of keep this infection at bay. Encased in these kind of tubercles, they can't really do much damage to the rest of your body, okay, which is why people at this stage, they might not show any symptoms. Okay? However, if you have a large infection, a few things can happen. That eventually, if you have too many of these tubercles, it's very damaging to the lung tissue, first of all, because then most of your, a significant amount of your lung tissue is now taken up or replaced with these tubercles. All right, and even though they're keeping the infection at bay, they very much are causing damage to the lung tissue. A, because they are occupying volume which could be used for gas exchange. They could be taking up, so we could be losing alveoli surface area, which is rather we'd like to be used for gas exchange, but instead we've got much of the volume of the lungs taken up by these tubercles. And secondly, these phagocytes, when they're moving, they are, remember, and we've done this before, they are releasing their digestive enzymes in order to kind of cut a way through the tissue to be able to get to the bacteria. So they are releasing digestive enzymes, these phagocytes, and the more the body has to, infuse this area with phagocytes to try and keep the infection at bay, the more they will be causing damage to the lung tissue by releasing their digestive enzymes. So this is very important, okay? So, two ways that you can be causing damage to the tissue, and remember this is ultimately what our discussion is about. The two ways are when you have many tubercles which are causing, taking up too much of the lung volume, and secondly, you have the phagocytes, large numbers of phagocytes in these lung tissues releasing digestive enzymes, ultimately causing damage to those tissues, damage to the integrity of that tissue. Okay, so remember these steps, right? Okay, so essentially you could say that the bacteria are causing a problem because they are attracting the immune cells. or the cells of the immune system, the lymphocytes and the phagocytes. Okay, so that's tuberculosis. I don't think you need to know this, but in very advanced stages, if these tubercles were to burst, and if these bacteria were to make it into the blood, they could go to all parts of the body, and essentially that's when the infection has reached a really dangerous stage, because once the bacteria escapes these tubercles and can go to all parts of the body, they can infect pretty much any part of the body and cause loss of functions in the organs that they infect. All right, okay, so that would be the advanced stage, but initial stages I think more relevant to you. All right, tuberculosis. Come back next and we'll talk, I think, about HIV. All right, guys, let's talk about HIV and how it can go on to cause AIDS. All right, so AIDS is the condition. Remember, HIV is the infecting particle. All right, the pathogen. Right, so remember, it's a virus. Now, I don't know why this is kind of skipped over in the textbook. Maybe you don't need to know this in that much detail, but I always find that kind of knowing things helps to understand, and understanding things helps to remember them. Yeah? Okay, so we have our HIV particle, all right? It's got a phospholipid membrane, and in the membrane it's got its viral proteins, which are very important, and inside it's got a single-stranded RNA genome. Remember, genome meaning the instructions to make more of that thing. Our genome is in our nucleus. It's double-stranded DNA. Okay, and as well as that, it's got a protein called reverse transcriptase. All right, so I hope you can see that. It's right there. Again, viruses are really, really small. All right, and in comparison, like, I don't know if this is exactly to scale, but remember we drew the bacterial cell really small compared to the eukaryotic cell. Well, a virus is, pretty much much smaller than a bacterial cell. So you could, an actual eukaryotic cell would basically go off the screen here. So that's kind of the scale of what we're talking about here. Anyway, let's go back to the situation here. How does HIV cause damage to its host? Essentially, that's what we want to know, because we're hoping that will make understanding everything else easier. So we have our virus and we have our human cell. All right, now actually HIV infects a particular type of human cell called the T helper cell. We encountered this cell in our section on immunity. It's the helper T cell, and it's responsible whenever the immune response is activated in response to a particular antigen. Helper cells is what the initial T cell differentiates into, as well as T killer cells and T memory cells, and the helper cells release the cytokines that promote the B cells to undergo their own expansion. Okay? So a pretty important cell in the immune response. So what we're thinking is, if we lose the function of this cell, we're pretty much saying, okay, we don't have an effective immune system. All right? So anyway, so this is our cell. It's also got a lipid bilayer membrane, just like the virus. And what happens is that through very specific interactions between the proteins on the outside of the virus and the proteins that happen to be on the outside of specifically helper T cells, there's a specific interaction there, and this interaction helps fusion of the membrane between the helper T cell and the virus itself. Remember, the virus had its own lipid-bilipid layer, and they fuse. And when they fuse, and they fuse because of the specific interaction, complementary interaction between viral protein and membrane protein on the helper T cell. Okay, so complementary binding interaction, whatever you want to call it, between the viral protein and the membrane protein. This is important, and complementarity between two proteins is a concept that runs all the way kind of through F212. So it's very important. Enzymes and substrates, inhibitors and enzymes, receptors and their ligands, hormones and receptors. Similarly, this, okay, the viral protein and the protein on the T helper cell surface membrane. They interact, and it's kind of like it initiates a response in the cell. Remember when we were talking in F211 about membranes, how sometimes when you lock with the right receptor, it makes the cell do something in response. Now in this case, it makes the cell fuse with that viral membrane and essentially let that thing in to the cell. So what happens is, now I'm just going to draw it bigger so you can see it. Now the viral RNA and the reverse transcriptase enzyme are both in the cytoplasm of the T helper cell. All right, so virus has entered the cell essentially. Okay, now what's going to happen is, just going to draw this here. Just going to draw that in your nucleus. In your nucleus, you have your DNA. All right, now I'm just going to draw effectively just one bit of it, just to show you what's going on. All right, now what happens is the reverse transcriptase, and this is why the reverse transcriptase is an essential component of the virus particle. The reverse transcriptase, it does what it says on the tin. It reverse transcribes. So remember, transcription is DNA copied into RNA. Reverse transcriptase, yes, my geniuses, reverse transcriptase copies the RNA into DNA. All right, so it will go along this strand and synthesize a DNA copy of that. And it will do it again, converting the single-stranded RNA genome into double-stranded DNA. Now this double-stranded DNA, I should have drawn it in a different color actually, this double-stranded DNA will now insert into the host genes, the host DNA. And what I will do, I'm just going to redraw this so it's easier to see. So the second strand, we'll draw it blue. So it copied the first strand, copy the RNA into DNA, and then copy the DNA again to make it double-stranded. Now what happens is that this viral genome, now converted into double-stranded DNA, inserts into the host genes. So makes a little cut, or there are enzymes to do this, cut in the right place, and suddenly the virus's genes have become part of the host's genome. This stage is called the provirus stage. And this is the reason why HIV is so difficult to A, detect, be detected by the host's immune systems, because at this stage there are no viral proteins, they're not virus particles around that you can detect as foreign. It's just DNA. It's just a sequence of bases in the DNA, whose, the cell can't really detect if genes belong to it or not. Okay, so that's why that's why the HIV particle is kind of difficult to be detected by the immune system, because it's most of the time in this kind of latent phase where there's no real symptoms. It's just sitting as part of the genes in the nucleus of the T helper cells, not really doing anything much. So when do the symptoms then come forward? When can you see the symptoms? When does it actually have an effect on the functioning of the host? That's the next stage. So shall we just recap then? Complementary interaction between viral protein and membrane protein on T helper cell. 2, RNA and reverse transcriptase enzyme enter cell. RNA is converted to double-stranded DNA and inserts into host cell genome. Okay, and we'll just call this, remember this, called this provirus stage. Okay, now what happens next? This is what actually causes damage to the T helper cells. Once the DNA has become part of this virus, and I'm not really sure when this starts to happen, but eventually it does start to happen, what happens is that the genes encoded in this DNA, the viral genes start to get transcribed and then translated into actual proteins. So viral genes get transcribed, i.e. messenger RNA. The ribosomes out here in the cytoplasm, they say, oh, look, there's some mRNA. Going to translate it, going to make some protein, right? So viral DNA gets transcribed and translated, and importantly, by host cell enzymes and ribosomes, and whatever kind of post-translational modification needs to be done. So that might even involve the Golgi. Okay, packing those proteins up for the virus, essentially. But whatever instructions are in the mRNA, that's what's being followed. So enzymes, ribosomes, Golgi, all of the host cell are being used to make viral proteins, producing viral proteins. All right. So once we get out here, we might find that there's going to be lots of copies of the original RNA. So as this gets copied, I hope you'll see we're making lots of copies of the original RNA. Okay? All right, so we'll have lots of copies of the original RNA. Those will be being translated by the ribosomes, and all the proteins that the virus had are encoded there. So we'll get lots of proteins, membrane proteins being produced. That's the spiky bits there. So they'll be being translated as these mRNAs are being read. And we'll also be producing, for the HIV, for new HIV particles, we'll also be producing lots of reverse transcriptase enzyme. Remember, it's a protein, and the instructions for that protein and this protein are on these mRNA strands, which are essentially copies of the viral genome in here. Okay, so as they are read by the ribosomes, so just draw a ribosome there, we're producing these proteins. All right. Now, These proteins will go through the Golgi body because it's a membrane protein, so it will, probably be formed on the endoplasmic reticulum or in the endoplasmic reticulum, sent to the Golgi body. The Golgi body will then package it up, any post-translational modifications, and send it off to the membrane. So they will end up here. Right? So these proteins will end up here. Okay? And we will have some of these, and we might just have our reverse transcriptase associating with it. Okay, so I hope you can see that everything to make more virus particles was essentially produced using the machinery of the host cell. And what will happen, I'm just going to redraw this. These particles will then bud off the cell, and as they do that, they will essentially be copies of what entered the cell in the first place. All right. Now when this happens, it can overwhelm the cell. So the cells, all the cell's resources are essentially being used to produce virus particles. There might not be enough nucleic acid, nucleotides, amino acids. There might not be enough of those around to then support the cell's own requirements. Remember, whenever you make something, it's an active process. Energy is used to build proteins, to build nucleic acid. And so this is also draining the energy of the cell. So, as we're producing lots of viral particles, it overwhelms the cell, and eventually it will cause the death of the cell because it doesn't have enough energy, it doesn't have enough resources for its own processes because it's being used up to produce these viral particles, okay? And eventually we will lose this T helper cell, and this process is happening in all T helper cells. So viral genes get transcribed, translated by the host. More virus particles are formed. Host cell overwhelmed, dies. Okay? And as we get this happening in many T helper cells, we effectively have no immune system, right? So when we do get, when we do get infected, even by cold or flu, or maybe even something more serious like tuberculosis, which normally might be kept at bay in an uninfected, HIV uninfected individual, because there's no immune system now to protect that individual, even opportunistic infections will then, prove very serious and probably will cause the death of HIV infected people. So that's when they've reached the acquired immunodeficiency syndrome stage. So they are immunodeficient, lacking in the immune system function, and therefore any infection, they cannot deal with it, okay, cannot be combated. All right, so host cell overwhelmed, dies, and then essentially the main point being opportunistic infections, e.g. tuberculosis, and there is a strong link here which I want you to remember. So opportunistic infections such as tuberculosis can now lead to death. Okay? All right, guys, so that's the process. That's HIV and AIDS. We'll come back and we'll look at malaria. Hi guys. You might not know this about me, but I am actually a very good artist. But unfortunately, because of time reasons, I've managed to produce this. Now, malaria, the life cycle it can be confusing, but if you break it down to its very most simplest, it's actually not too difficult. Hopefully, I will convince you of this in the next few minutes. Okay, but what we're talking about here is how is the transmission part that's difficult to understand. So let's just begin. Now, malaria, remember I said that it needs a vector. It can't go from person A to person B directly, okay? Cannot happen. It needs a vector, and that vector is a mosquito, all right? The female Anopheles mosquito, right? So let's just, the very most basic thing that you must know is that it can only go from person A to person B via the mosquito, all right? Mosquito bites infected person, takes up the pathogen into its own body, and then the pathogen, when it bites a second uninfected person, the pathogen then passes into person B. At its very basic, that's the idea. If you want to look in a little bit more detail, that can also be done, all right? So, let us begin with this mosquito, all right? Now, this mosquito has bitten an infected person, okay? And it has the Plasmodium, the infective stage of the Plasmodium in its salivary glands. So, infective stage of Plasmodium, so that's the protoctist. It goes through a number of stages in its life cycle, and that's what's being shown here, right? So, infective stage of Plasmodium is in salivary glands of this mosquito, right? So what we'll do is maybe just show where the Plasmodium is with red, okay? So the red right now is in the salivary glands of this mosquito. So when it bites, pierces the skin of this individual, inserts the proboscis into, one of the capillaries, at that point it injects a little bit of its saliva into that region so that the blood doesn't clot and the mosquito doesn't get essentially stuck there, okay? So, anticoagulant going in, but with the anticoagulant, the Plasmodium then enters the bloodstream of this individual. Okay? So now this person is infected, right? So, infective stage of the Plasmodium from salivary glands into the blood of this person. From the blood, it will then go to the liver, okay, where it multiplies. And I'm just giving you the essentials here, so I suggest that you make a note of this. So infective stage from the salivary glands into the blood to the liver, multiplies, and from there migrates to blood cells. Okay, now it's this effect on the blood cells. So they multiply a lot in the blood cells, and they can cause the red blood cells to burst, lowering, it causes fever as the body puts lots of energy into then kind of producing more red blood cells. So this, the fever that's produced by malaria is because the body is putting a lot of its energy into repopulating its red blood cells in the blood. Okay, so when these when the plasmodium gets into the red blood cells, they multiply a lot and cause the red blood cells to burst. Okay. And whenever someone has malaria but they're not showing any symptoms of it, that's a latent stage in the liver. Okay, so let's just make a note of that. There can be a latent stage in liver where they've not migrated to the blood cells, they're just in there. But when they do migrate to the blood cells, they rapidly multiply, causing red blood cells to burst, and that's essentially what the kind of damage is to the person. Okay. But another thing also happens. Why is this person here? Anyway. So, what happens next is, in the blood, they form the gametes. Okay, so in the blood cells, you can think of meiosis happening here. All right, so in the plasmodium, gametes are formed. All right, so I've just indicated here them as symbols, male and female. So female being this symbol, so you can think of it as the female gamete, and the male gamete given this symbol. Okay, so these gametes are being formed also in the red blood cells. Now, what can happen is, so these, in the red blood cells, we've got the male and female gametes of the plasmodium. Remember, we need them to come together in order to form a kind of infective stage plasmodium, right? Right now it's just gametes. So what happens here is, if mosquito comes along, doesn't have any plasmodium in its system, so so we talked about mosquito number 1. Now we are talking about mosquito number 2. So second mosquito arrives and bites this infected person. Remember, the plasmodium has gone in this person's liver. It's then moved into his or her red blood cells, and in the red blood cells they form the gametes. And when this mosquito bites this person, it will take up those gametes, both the male and the female gametes, and they will go into its stomach. No idea where a mosquito's stomach is, but I'm guessing it's somewhere around here. Okay? Now those gametes make it into the stomach, and here they fuse. Here they fuse, forming infective stage plasmodium again. And it's these that then move into the salivary glands, and then when this mosquito bites second person, the whole thing will start again. Okay? I think that's pretty clear. Shall we do it again one more time? Right? So, mosquito, remember, now let's just continue the story then. So when this person, sorry, not person, but when this mosquito, now remember, it bit the infected person, and now there's going to be an uninfected person again. Now this mosquito, it bit the person that was infected. They had gametes in their red blood cells. The red blood cells obviously got sucked up by the mosquito into the stomach. The gametes fused, forming the infective stage plasmodium again, which resides now in the salivary glands. When this mosquito bites another uninfected person, then again the plasmodium, the infective stage plasmodium, will go out of the proboscis into the blood cell of the uninfected person, to their liver. Now they're infected, to their liver, multiply here, migrate to the red blood cells. In the red blood cells, they will form gametes again, and so on. All right, and then when another mosquito comes along and bites this person, and then maybe bites another person, it will have transmitted it to the third person. Okay, and so the cycle requires both the mosquito and the human. Okay, right? So remember, if there's any symptoms caused in the individual, it's because they're losing their red blood cells, because the plasmodium is multiplying and causing them to burst. But if that doesn't happen, you know, essentially it's just the fact that they are in the red blood cells, the gametes are there, and the gametes will fuse to form the zygote. Forgot to mention that, right? Fuse to form the zygote. Maybe you should call it infective stage. Let's just call it fuse to form the zygote in the stomach, and infective stage, then when they mature and they move to the salivary glands, that's when we have the infective stage. All right. Okay, so that's malaria. I think that's it, guys. I think I'm not going to have any more time to make more videos, so that's it. Remember, if you've got questions on anything, please do you know, leave a comment on any of the videos, and I will try and respond to you as soon as I can. But I think time has beaten me in terms of making more videos, so I will wish you good luck. You can do it. Keep working, keep asking questions, keep practicing papers, looking at mark scheme, doing the questions again, and you'll get there.